Microbiology Chapter 18, Adaptive Specific Host Defenses. The immune system serves as the body's defense mechanism against harmful pathogens. Innate immunity is the first line of defense, providing immediate non-specific responses to infections. This includes physical and chemical defenses such as the skin and the mucous membranes, as well as immune cells that recognize and attack invaders. In contrast, adaptive immunity develops over time, offering a tailored response to specific pathogens. It involves lymphocytes such as B cells and T cells, which remember past infections to mount stronger attack upon re-exposure. Together, these two systems work harmoniously to protect the body from disease and maintain health. The slide illustrates a pretty detailed summary of the Chapter 17, Innate Non-Specific Host Defenses, and this Chapter 18, Adaptive Specific Host Defenses. People born in the 1960s or later in developed nations may struggle to grasp the significant impact of infectious diseases that once posed severe threats. For instance, smallpox, a lethal viral disease, devastated populations until its eradication through vaccination efforts led by organizations like the World Health Organization, Rotary International, and UNICEF. Since 1977, no cases of smallpox have been reported. Similarly, polio, which caused paralysis and required patients to rely on the iron lung wards until 1950s, has nearly been eradicated due to vaccination initiatives. Vaccines have also reduced the prevalence of once common infectious diseases such as chickenpox, German measles, mumps, and whooping cough. The success of these and the other vaccine is due to the very specific and adaptive host defenses that are the focus of this chapter. Innate non-specific host defenses describe innate immunity against microbial pathogens discussed in the previous chapter. Higher animals such as humans also possesses an adaptive immune defense which is highly specific for individual microbial pathogens. This specific adaptive immunity is acquired through active infection or vaccination and serves as an important defense against pathogens that evade the defenses of innate immunity. The chapter outlines the key concepts in immunology, beginning with the definition of memory, primary response and secondary response, and specificity, which are crucial for understanding how the immune system remembers the past infections. It differentiates between the humoral immunity, which involves antibodies produced by the B cells, and the cellular immunity, which is mediated by the T cells. Additionally, it clarifies the distinctions among antigens, epitopes, and haptens, emphasizing their role in immune recognition. Finally, the text describes the structure and function of antibodies, detailing the various classes of antibodies and their specific roles in the immune response. Adaptive immunity is characterized by two key features, specificity and memory. Specificity refers to the immune system's ability to target particular pathogens, while memory enables it to respond rapidly to previously encountered pathogen. For instance, after recovering from chickenpox, the body retains some memory of the varicella zoster virus, ensuring protection against future infections by the same virus. These features are facilitated by the programming of certain immune cells that respond quickly upon subsequent exposure to the same pathogen. This programming starts with a primary response triggered by an initial encounter with a pathogen or vaccine. When the body faces the pathogen again, it generates a secondary response that is both faster and more robust due to the pre-existing memory. However, this response is specific to the original pathogen, meaning that immunity to one virus, such as varicella zoster virus, does not confer protection against other viral infections like measles, mumps, or polio. Adaptive specific immunity involves the action of two distinct cell types, the B lymphocytes, the B cells, and the T lymphocytes, or the T cells. Although B cells and the T cells arise from the common hematopoietic stem cells differentiation pathway, their sites of maturation and their role in adaptive immunity are very different. B cells will mature in the bone marrow, produce antibodies or immunoglobulins essential for defending the body against pathogen and toxins in the extracellular environment, a process known as humoral immunity. In contrast, the T cells mature in the thymus and act as central coordinators of both innate and adaptive immune responses, specifically targeting and destroying cells infected with intracellular pathogens through cell-mediated immunity. Activation of adaptive immune defenses is initiated by specific molecular structure known as antigens. While antigens share similarities with pathogen-associated molecular pattern or PAMPs that we discussed, that are found across many pathogens, they are unique to individual pathogens. For instance, the antigens responsible for triggering adaptive immunity to chickenpox are specific to varicella zoster virus and distinctly differ from those associated with other viral infections. The term antigen originally referred to molecules that induce antibody production, derived from the combination of antibody and generator. A molecule that stimulates antibody production is described as antigenic. However, antigens are not solely linked to humoral immunity, they also play a critical role in activating cellular immunity, leading to the more precise term of immunogens. In this discussion, we will primarily use the term antigens. Various pathogen structures that can harbor antigens, for example, bacterial antigens may be linked to their capsules, cell walls, fimbriae, flagella, or pili. Additionally, these antigens can be associated with toxin and enzymes secreted by the bacteria. In the case of viruses, antigens are found on their capsids, envelopes, and specific structures that facilitate cellular attachment. Antigens are diverse molecules that can belong to various classes such as carbohydrates, lipids, nucleic acid, and proteins. Their effectiveness in stimulating adaptive immune responses varies significantly across these classes, with the structural complexity of antigenic molecule being a key factor in its potential. Generally, more complex molecules, particularly proteins, are more effective at eliciting both humoral and cellular immunity. In contrast, carbohydrates are less complex and can primarily stimulate humoral defenses, while lipids and nucleic acids are least antigenic unless they are combined with proteins or carbohydrates to form structures like glycolipid and lipoproteins. The three-dimensional complexity of antigen is crucial because antibodies and the T cells interact with specific regions on the antigens known as epitopes, rather than the entire antigen itself. A single antigen can have multiple epitopes, allowing different antibodies to bind to different sites. For instance, the bacterial flagellin, which is a large protein structure, can contain hundreds or thousands of unique epitopes that are specifically recognized by antibodies. This specificity can vary even among the flagella from different bacterial species or strains, underscoring the importance of epitope diversity in immune responses. Size is another critical factor affecting the antigen's potential. While large antigens have multiple epitopes, some smaller molecules termed haptens are not antigenic on their own. To elicit an immune response, haptens must attach to a larger carrier molecule to form a conjugate or antigen. The immune system can then produce hapten-specific antibodies that recognize both the conjugate and the free hapten. Although haptens are not linked to specific pathogens, they can trigger allergic reactions, as seen is urushiol, poison, and penicillin, which can cause immune responses leading to symptoms like contact dermatitis. Antibodies, also known as immunoglobulins, are glycoproteins found in the blood and the tissue fluids. The basic structure consists of 4 protein chains linked by a disulfide bond, which are covalent connection between the sulfhydryl and the R group of 2-cysteine amino acid. The 2 largest state chains are identical and referred to as heavy chains, while the 2 smaller chains known as the light chains. Together, these chains create a Y-shaped structure, which is essential for the antibody's function. The Y-shaped antibody features the arms known as the FAB or the Fab region, short for fragment of the antigen binding, where the far end contains a variable region that binds to the specific antigens. The amino acid sequences in this variable region determines the three-dimensional structure, allowing the Fab region to interact with unique epitopes. Although the epitope specificity is the same for both arms of the single antibody, there is significant variability among different antibodies. This binding capability is crucial for neutralizing pathogens, facilitating agglutination or aggregation of pathogens, and enabling antibody-dependent cell-mediated cytotoxicity. The constant region of the antibody molecule includes the trunk of the Y or the lower portion of each arm of the Y. The trunk of the Y is also known as the FC region for fragment of crystallization and is the site of complement factor binding and binding to the phagocytic cells during antibody-mediated opsonization. The constant region of the antibody molecule is crucial as it determines its class or isotype of antibody. There are 5 classes of antibodies: IgG, IgM, IgA or IgD, and IgE. Each class has distinct heavy chains identified by Greek letters: gamma for IgG, mu for IgM, alpha for IgA, delta for IgD, and epsilon for IgE. These classes differ significantly in their abundance in the serum, structural arrangement, site of action in the body, functional roles, and overall size. IgG is the most prevalent antibody in the human blood, comprising 80% of the total serum antibodies. It exists as a monomer and is highly efficient in penetrating tissue spaces. Notably, IgG is the only antibody class that can cross the placental barrier, providing passive immunity to the fetus during pregnancy. Its versatility makes the immunoglobulin G or IgG a critical component of the body's defense mechanism against various pathogens. IgM is produced as a monomer that acts as an antigen-binding receptor on B cells. When secreted, IgM forms a pentamer consisting of 5 monomers connected by a J chain, which limits some of its function compared to the IgG. However, the 10 Fab or the Fab sites available from the pentameric IgM contributes significantly to its importance in immune responses. IgM is the first antibody produced during both primary and secondary immune responses, making it a valuable diagnostic marker for recent infections. Accounting for about 13% of the total serum antibodies, IgA is primarily found in mucus secretions and protect mucous membranes. It also appears in other body fluids including breast milk, tears, and saliva. Secretory IgA is typically in a dimeric form with 2 monomers linked by a secretory component. One of the key functions is to trap the pathogens in the mucus, facilitating their elimination from the body. The IgD serves as the membrane-bound monomer on B cells and act as an antigen-binding receptor. However, it is not secreted and exists in only trace amount in serum, likely due to the degradation of older B cells. The presence of IgD is primarily associated with the surface of the B cell rather than the systemic immune responses. Finally, the IgE is the least abundant class of antibody in the serum and secreted as a monomer. Its role in adaptive immunity is primarily focused on antiparasitic defenses. IgE binds basophil and mast cells, where its Fab region interacts with the specific antigen epitopes, leading to the release of pro-inflammatory mediators. This inflammatory response is essential for combating parasites, but it also plays a significant role in allergic reactions. Different classes of antibodies play crucial roles in the body's defense against pathogens. Their functions include neutralization of pathogen, opsonization for phagocytosis, agglutination, complement activation, and antibody-dependent cell-mediated cytotoxicity. Antibodies serve as a vital link between the adaptive specific immunity and innate nonspecific immunity, ensuring a coordinated response to infections. Neutralization occurs when antibodies such as IgG, IgM, IgA bind to epitopes on pathogens or toxin, effectively preventing their attachment to host cells. For instance, secretory IgA can attach to specific pathogens, blocking their initial interaction with intestinal mucosal cell. This mechanism also applies to toxin, wherein the antibodies can prevent them from binding to target cells, thereby neutralizing their harmful effects. Additionally, viruses can be neutralized in this manner, stopping them from infecting healthy cells. Opsonization refers to the process of coating pathogens with molecules like complement factors and specific proteins to enhance phagocyte binding, facilitating phagocytosis. IgG antibodies are particularly effective as opsonins as they bind to specific epitopes on pathogen surface through their FAB sites. Phagocytic cells, including macrophages and the neutrophils, recognize the Fc portion of this IgG, allowing these immunocells to attach and engulf the pathogen with the antibodies. Agglutination is the process by which the antibodies actually cross-link the pathogen, forming a large aggregate that are easier for the immune system to eliminate. IgG, with its two FAB antigen binding sites, can bind to separate pathogen cells, leading to clumping. When multiple IgG antibodies are involved, significant aggregates form, which are more readily filtered by kidneys and the spleen. The pentameric structure of IgM enhances this process, providing the 10 Fab binding sites per molecule, making it the most efficient antibody for agglutination. Antibodies play a crucial role in the activation of the complement cascade, which is a key component of the innate immune response as discussed in the previous chapter. This system enhances the inflammation, recruits phagocytes to infection site, promotes phagocytosis through opsonization, and destroys gram-negative bacteria using the membrane attack complex, or the MAC. Activation of the complement system occurs through three distinct pathways, classical pathway being the most effective, requiring the IgG or the IgM antibodies to bind to the pathogen surfaces and the recruitment of the C1 complement complex. Additionally, antibodies are involved in antibody-dependent cell-mediated cytotoxicity, or ADCC, which facilitates the destruction of larger pathogens that cannot be phagocytosed. The process is primarily associated with natural killer cells, but also includes macrophages and eosinophils. During ADCC, the Fab region of the IgG antibody attaches to the large pathogen, while the Fc receptor on effector cells bind to the antibody's Fc region. This interaction positions the effector cells near the target, allowing it to release the cytotoxic molecules such as perforins or granzymes to eliminate the pathogen effectively. In this section, identification of cells that express major histocompatibility complex, or MHC1 and 2 molecules, detailing their structure and cellular locations, will be discussed. The discussion will also include the role of antigen-presenting cells, which include dendritic cells, macrophages, and B cells in the immune response. Additionally, the section will describe the process of antigen processing and presentation for both MHC1 and MHC2, highlighting how these molecules present antigens to T cells, thereby initiating the adaptive immune responses. The major histocompatibility complex, or MHC, is the vital group of genes responsible for producing MHC molecules located on the surface of all nucleated cells in the human body. In humans, these genes are commonly referred to as human leukocyte antigen (HLA) genes. The only exception to this rule is the mature red blood cells, which do not have a nucleus and therefore do not express MHC molecules. MHC molecules play a critical role in adaptive immunity and are classified into two main types, MHC1 and MHC2. MHC1 molecules are present on all nucleated cells and are involved in presenting both normal self-antigens and abnormal or non-self pathogens to effector T cells, which are crucial for cellular immunity. On the other hand, MHC2 molecules are restricted to specific immune cells such as macrophages, dendritic cells, and B cells, and they function by presenting abnormal or non-self pathogens to antigens for initial activation of T cells. Structurally, MHC1 and MHC2 molecules differ significantly despite both being transmembrane glycoproteins that form dimers in the cytoplasmic membrane. MHC1 is made up of a longer protein chain paired with a smaller beta-2 microglobulin, which only the alpha chain spanning the membranes. In contrast, MHC2 consists of two protein chains, an alpha and a beta chain, which are similar in length, both of which span the plasma membrane. The antigen binding site of the MHC molecule, which is essential for presenting antigens to T cell, is located near the top of the dimer. Specifically, the MHC1's cleft is formed by the alpha-1 and the alpha-2 domains, while the MHC2's cleft is created by the alpha-1 and the beta-1 domains. The self-explanatory slide compares the MHC1 and MHC2 molecules. It is important to note that the CD8 co-receptor of a cytotoxic T cell binds to the alpha-3 region of the MHC1 molecule. In contrast, the CD4 co-receptor of the helper T cells bind to the beta-2 region of the MHC2 molecule. In addition, MHC1 can accommodate a smaller peptide of about 8 to 11 residues, whereas MHC2 can present a larger peptide, about 13 to 25 residues, to the helper T cells. These details of the MHCs and the T cell interaction will be discussed shortly in the subsequent slide in more detail. All nucleated cells in the body can process and present antigens alongside MHC molecules, which signals the immune system about the status of the cell, whether it is healthy or infected by an intracellular pathogen. These cells are called the non-professional antigen-presenting cells. However, only specific cells, namely macrophages, dendritic cells, and the B cells, can present antigens to activate T cells, categorizing them as professional antigen-presenting cells, or APCs. While the APCs contribute to adaptive immunity, they have distinct functions. Macrophages and dendritic cells can act as phagocytes, engulfing and destroying pathogens that breach the body's initial defenses, such as the skin and the mucous membranes. In contrast, the B cells do not ingest pathogens. Instead, they are primarily responsible for producing and secreting antibodies. Furthermore, macrophages and dendritic cells recognize pathogens through nonspecific interaction with various receptors, while B cells utilize antigen-specific immunoglobulin receptors, monomeric IgD and IgM, to bind to and internalize antigens via endocytosis before presenting them to T cells. MHC2 molecules are exclusively found on the surface of antigen-presenting cells, or APCs, such as macrophages, dendritic cells, which utilize similar processes for antigen processing and presentation. B cells employ a different mechanism, which will be covered in relation to the B lymphocyte and the humoral immunity. The current focus is on the steps involved in antigen processing, specifically for dendritic cells. When a dendritic cell identifies and attaches to a pathogen, it internalizes the pathogen through phagocytosis, initially containing it within a phagosome. This phagosome then fuses with the lysosomes containing antimicrobial enzymes, forming a phagolysosome where the degradation of the pathogen begins. Class II MHC molecules bind peptides derived from exogenous antigens internalized by the phagocytosis or endocytosis and processed within the endocytic pathway. APCs can internalize antigens by phagocytosis and or endocytosis. Macrophages do both. B cell-used receptor-mediated endocytosis. It is important to note that APCs do not present every possible epitope to the T cells. Rather, they selectively present the most immunogenic or dominant epitopes. The selection mechanism for processing and presentation of these epitopes by an APC is complex and not fully understood. Once the most antigenic epitopes are processed, they bind within the antigen binding cleft of the MHC2 molecule and are transported to the dendritic cell surface for presentation to the T cells. MHC1 molecules are present on all healthy nucleated cells and serve to signal to the immune system that these cells are normal self-entities. In a healthy cell, cytoplasmic proteins are degraded by proteasomes into self-antigen epitopes, which then bind to MHC1 molecules and are presented on the cell surface via endogenous pathway. Immune cells, such as natural killer cells, recognize these self-antigens and refrain from attacking cells. The endogenous processing pathway involves class 1 MHC molecules binding peptides derived from endogenous antigen processed within the cytoplasm. Proteins can enter the cytoplasm from phagocytosed microbes via endogenous synthesis, such as from the viruses in infected cells. These proteins are unfolded, ubiquitinated, and degraded by proteasomes into smaller peptides. The resulting peptides are then transported into endoplasmic reticulum by the TAP transporter, where they bind to the newly synthesized class 1 MHC molecules, specifically those with 8 to 11 amino acid residues. These MHC class 1 antigen complex then migrate to the cell surface to be presented to the CD8 plus T cells, which can recognize the complex and initiate further immune processing. For the elimination of the infected cells to proceed, antigen-presenting cells must activate the T cell involved in the cellular immunity. If an intracellular pathogen infects the APC directly, it can process and present antigens via MHC1 as described. Conversely, if the pathogen does not directly infect the APC, it employs a mechanism called the cross-presentation. In this process, the antigens are internalized by the APC through methods associated with MHC2 presentation, such as phagocytosis, but are then displaced on MHC1 molecules for recognition by a CD8 cells. Although the mechanism of cross-presentation are not fully understood, it is primarily attributed to the dendritic cells rather than macrophages or the B cells. In this section, the T cell maturation and the thymic selection will be discussed. In addition, T cell diversity generation, subtypes of major T cells, and the role of superantigens will also be discussed. Humoral immunity primarily targets extracellular pathogens and toxins by using antibodies to prevent their attachment to host cells, while cellular immunity focuses on eliminating intracellular pathogens through T lymphocytes or T cells. T cells are crucial not only for combating intracellular threats, but also for coordinating the overall adaptive immune response, which includes both humoral and cellular defenses, as well as the innate immune responses. T cells, essential components of the immune systems, derive from the multipotent hematopoietic stem cells, or HSCs, in the bone marrow. Unlike white blood cells of innate immunity, T cells first differentiate into lymphoid stem cells and then into small immature lymphocyte known as lymphoblasts. This initial differentiation occurs in the red marrow of the bones, after which these immature T lymphocytes enter the bloodstream and travel to the thymus for final maturation. Upon arrival at the thymus, they are termed as thymocytes. The maturation of thymocytes in the thymus encompasses 3 critical steps, collectively known as thymic selection. The first step occurs in the thymic cortex, where thymocytes develop functional T cell receptors, or TCRs, necessary for activation by antigen-presenting cells. Any thymocyte with defective TCRs are eliminated through negative selection, which involves apoptosis. The second step, also in the cortex, focuses on positive selection, where thymocytes that successfully interact with MHC molecules receive stimulation to progress further in maturation, while those that do not interact properly are removed. The third and the final step of thymic selection takes place in both the cortex and the medulla, involving negative selection to eliminate self-reacting thymocytes. This process is crucial for establishing central tolerance, preventing potentially harmful self-reacting T cells from entering the bloodstream that cause autoimmune diseases. Despite these, some self-reactive T cells may evade this selection and enter peripheral circulation, necessitating additional mechanisms to prevent autoimmune responses. To manage the self-reactive T cells that escape the thymus, peripheral tolerance mechanisms come into play. This includes anergy, a state of responsiveness to antigen stimulation, which occurs when self-reactive T cells lack essential co-stimulatory signals for activation. Additionally, regulatory T cells play a significant role in peripheral tolerance by inhibiting self-reactive T cells and secreting anti-inflammatory cytokines. These regulatory T cells may develop during the thymic selection process, possibly receiving unique signals that help them avoid negative selection. Ultimately, thymic selection is highly effective, eliminating approximately 98% of the thymocytes. The remaining 2% that successfully exit the thymus migrate through the bloodstream and lymphatic system to secondary lymphoid organs, such as lymph nodes and the spleen. These surviving T cells are known as the mature naive T cells, remaining dormant until activated by specific antigens presented by the APCs, thereby playing a pivotal role in adaptive immunity. T cells are integral components of the adaptive immune system and can be classified into three main types: helper T cells, regulatory T cells, and cytotoxic T cells. The differentiation among these classes is based on the expression of specific surface molecules, their activation processes, and their unique roles in immune responses. Each classes of T cells play a vital role in maintaining immune function and homeostasis. All T cells express cluster of differentiation or CD molecules, which are glycoproteins on the cell surface that help identify and differentiate among various white blood cells. The most significant CD molecules for the T cell classifications are the CD4 and CD8. Helper T cells and regulatory T cells are distinguished by the presence of CD4, while cytotoxic T cells are marked by CD8. This distinction is crucial for understanding their respective functions in the immune system. The activation of the T cells also relies on the interaction of the major histocompatibility complex molecules and antigen-presenting cells. Helper and regulatory T cells are activated by the APCs presenting antigens linked to MHC2, while cytotoxic T cells recognize antigen presented by MHC1. This difference in activation pathways underscores the distinct roles of each T cell class plays in response to infections and managing immune responses. Functionally, each class of T cell has a unique role in the immune systems. Helper T cells act as central coordinators, facilitating the activation of both humoral and cellular immunity, but also enhancing the pathogen-fighting abilities of the innate immune cells like macrophages and natural killer cells. Regulatory T cells are crucial for maintaining the immune tolerance and preventing autoimmune diseases. In contrast, the cytotoxic T cells serve as the primary effectors in cellular immunity, targeting and destroying the cells that are infected with intracellular pathogens. Together, these T cell classes contribute significantly to the body's defense mechanism. Activation of the helper T cells and cytotoxic T cells is a multifaceted process that relies on the interplay of various molecules and influence of cytokines. Central to this activation is the T cell receptor or TCR, which plays a pivotal role in recognizing the pathogen epitopes. The TCR belongs to the same family such as immunoglobulins, specifically IgD and IgM, which serves as antigen receptors on the B cells. While TCR antibodies share a common structural features, the TC is comparatively smaller and simpler, consisting of two peptide chains, alpha and beta, that span the T cell's cytoplasmic membrane. TCRs are uniquely epitope-specific, with estimates suggesting that an individual may require around 25 million distinct T cells, each with a unique T cell receptor to effectively defend against various microbial pathogens. Given that the human genome contains only 25,000 genes, this raises an intriguing question of how such a vast repertoire of TCR can be generated. The answer lies in a genetic rearrangement process that occurs in the thymus, which is crucial during the initial phase of thymic selection. The genetic blueprint for TCR variable regions is organized into gene segments categorized as variable V, diversity D, and joining J segments. Specifically, the alpha chain of the TCR incorporates over 70 different V alpha segments and 61 J alpha segments, while the beta chain consists of 52 V beta segments, 2 D beta segments, and 13 J beta segments. This organization allows for a high degree of combinatorial diversity during TCR development. As T cells mature in the thymus, genetic rearrangement brings together the specific segments to construct the variable regions of the TCR. For the alpha chain, one V alpha segment is paired with one J alpha, while the beta chain is formed by combining V beta statement with a D beta and the J beta segment. The extensive combinations made possible through these rearrangements yield the necessary genetic diversity to produce millions of TCRs, each specifically tailored to recognize distinct epitopes. Helper T cells are crucial players in the immune response, requiring activation by antigen-presenting cells that display processed foreign epitopes alongside MHC class II molecules. The activation process begins with the T cell receptors identifying a specific epitope within the MHC2 antigen binding cleft. Following this recognition, CD4 on the helper T cell interacts with a distinct region of the MHC2 molecule, stabilizing the complex and ensuring that the T cell acknowledges both the foreign epitope and the self-antigen of the APC. This dual recognition is essential for the full activation of the helper T cell. Once activated, the helper T cell undergoes a proliferation phase, dividing through mitosis and generate clonal population of naive helper T cells. These cells can differentiate into distinct subtypes, a process guided by the cytokines secreted by the APCs. The activated helper T cells may evolve into T helper Th1, T helper 2 Th2 cells, and the memory helper T cells. Th1 and Th2 cells function as short-lived effector cells, providing immediate responses, while memory helper T cells are long-lived and crucial for rapid reactions to subsequent exposure to specific antigens. Th1 cells are pivotal in coordinating the broader immune response by secreting cytokines that enhance the activity of other immune cells. They stimulate cytotoxic T cells, improving their ability to eliminate infected cells and promoting their differentiation into memory cytotoxic T cells. Additionally, Th1 cells activate macrophages and neutrophils, enhancing their effectiveness against intracellular bacteria, and can also empower natural killer cells in their target cell destruction. Conversely, Th2 cells are integral to the humoral immune response. They secrete a variety of cytokines that activate B cells, guiding their differentiation and facilitating the production of antibodies. Th2 cells orchestrate the process of antibody class switching, enabling B cells to produce different classes of antibodies tailored to specific immune challenges, thus enhancing the overall effectiveness of the humoral response against pathogens. A more recently identified subtype, Th17 cell, has emerged as significant in defending against the chronic mucocutaneous infections. Their specific cytokines are vital for immune responses that are not adequately addressed by the Th1 or Th2 cells. Patients with deficiencies in the Th17 cells, such as those with HIV, may face increased susceptibility to severe infections, underscoring the importance of this subtype in maintaining mucosal immunity and overall immune health. As mentioned previously, memory helper T cells are relatively long-lived and are programmed to remember a specific antigen or epitope in order to mount a rapid, strong secondary response to subsequent exposures. Cytotoxic T cells, also known as cytotoxic T lymphocyte or CTLs, are crucial components of the immune response activated through a three-step process involving antigen-presenting cells. The activation differs from that of helper T cells, as a cytotoxic T cell recognize antigen presented with MHC1 molecules and engage with CD8 instead of CD4. Following the successful interaction between the T cell receptor and the foreign epitope, along with the self-antigen, cytokines produced by both the APC and the cytotoxic T cell stimulate clonal proliferation and differentiation. This process results in the formation of effector cytotoxic T cell, which target and destroy pathogen, as well as memory cells that prepare for future encounters. The proliferation and differentiation of cytotoxic T cells are further enhanced by cytokines released from the Th1 cells that have also been activated by the same foreign epitope. While cytotoxic T cell activation can occur independently of the Th1 cells, this auxiliary stimulation significantly improves the effectiveness of longevity and the immune response. The synergy between the Th1 cells and the cytotoxic T cells underscores the complexity of the adaptive immune system, where the multiple cell types work together to enhance protective mechanism against pathogens. Once activated, cytotoxic T functions as the primary effector cells of cellular immunity, specifically targeting and eliminating cells infected with intracellular pathogens. Unlike natural killer cells or NK cells that respond to the non-specific signal of cell stress, cytotoxic T cells specifically recognize infected cells through the presentation of pathogen-specific epitopes associated with MHC1. Upon recognizing an infected cell, the cytotoxic T cells TCR bind to epitope, triggering the release of perforin and granzymes. Perforin forms pore in the infected cell membrane, allowing granzymes proteases to enter and induce apoptosis. This careful controlled mechanism ensures that the infected cells are efficiently destroyed without releasing pathogens, thereby preventing the spread of the infection to adjacent healthy cells. T-cell activation is crucial for effective immune response against infections, but when this activation becomes uncontrolled, it can lead to severe consequences. Certain pathogens, particularly some bacteria and viruses, produced toxins known as superantigens that can cause this dysregulated response. Notable bacterial superantigens include toxic shock syndrome toxin, TSST, staphylococcal enterotoxins, and various streptococcal toxins. Additionally, viruses such as Epstein-Barr virus, cytomegalovirus also produce superantigens. The mechanism by which superantigens activate T cell involves their ability to stimulate bind to MHC2 molecules on an antigen-presenting cells and variable region of the T cell receptor beta chain. This interaction occurs outside the typical antigen binding cleft of MHC2, allowing for binding of MHC2 and TCR without the need for specific epitope recognition. Consequently, these lead to a massive release of cytokine, resulting in a cytokine storm that can cause a severe inflammatory response, leading to critical conditions such as decreased blood pressure, shock, multi-organ failure, and in extreme cases, death. Collectively, this phenomenon is known as immunopathology. The immune system is good at protecting us every day, but it can kill us too if it malfunctions in certain situations like these. This section will discuss the production and maturation of the B cells. The section will also compare the T cell and the B cell receptor, along with the activation of the B cell by two different mechanisms, T-dependent and T-independent activation. In the primary and secondary antibody responses will be also discussed. B cells originate from the hematopoietic stem cells in the bone marrow, progressing through several developmental stages. Initially, the HSCs differentiate into multipotent progenitor or MPP cells into common lymphoid progenitors or SCLAP cells or CLP cells. As B cells develop, they undergo the VDJ recombination as mentioned before, rearranging the immunoglobulin H and L chain gene loci, which is essential for their function. During the development in the bone marrow, B cells face two selection processes to ensure their proper maturation. Positive selection involves the pre-B cell receptor and the B cell receptor signaling without the presence of antigen. If these receptors fail to bind their ligands, the B cells do not receive necessary signals and stop developing. Negative selection occurs when the BCR binds strongly to self-antigens, leading to several possible outcomes, including clonal deletion or receptor editing. This process establishes central tolerance, ensuring that mature B cells do not react to self-antigens. Once immature B cells complete their development in the bone marrow, they migrate to the spleen as transitional B cells, initially referred to as T1 B cells. Upon entering the spleen, they transition into the T2 B cells, ultimately differentiating into follicular B cells, marginal zone MZ B cells, depending on the signals received. These differentiated cells are classified as mature or naive B cells, marking the final stage of their development. Germinal centers are temporary structures located in the B cell zone of secondary lymphoid organs, such as lymph nodes, Pierce patches, and the spleen, where mature B cells undergo activation, proliferation, differentiation, and somatic hypermutation of their antibody genes to enhance affinity during an immune response. Most of the B cells formed in these centers are eventually eliminated by the tangible body macrophages. B cells, like the T cells, are equipped with antigen-specific receptors that exhibit diverse specificities. While they typically require T cell assistance for optimal function, B cells can also activate it independently. Naive mature B cells express B cell receptors that are membrane-bound forms of immunoglobulin D and M, IgM. These receptors consist of two identical heavy chains and two identical light chains arranged in a Y shape with a constant region integrating into B cell membrane. The exposed antigen binding sites are crucial for recognizing specific pathogen epitopes, initiating the activation of the B cells. Each naive B cell can present around 100,000 identical BCR on its surface, all targeting the same epitope. To effectively respond to the broad array of microbial epitopes, B cells undergoes the genetic rearrangement similar to the T cells, creating a diverse range of receptor specificities. The variable region of the BCR's heavy chain is constructed from the various genes of the VDJ, akin to the beta chain of the T cell receptors. The light chain's variable region comprising the V and the G segments mirroring the alpha chain of the TCRs. This genetic rearrangement allows for millions of unique antigen binding sites, enhancing the BCR and the antibodies produced following B cell activation. The Ig alpha protein, a component of the B cell receptor containing an ITAM, which is immunoreceptor of tyrosine-based activation motif. This item is crucial for initiating B cell signaling upon antigen recognition. The Ig alpha item, along with the item of the Ig beta protein, which is also the part of the BCR, is phosphorylated by Circ family kinases, which then activates downstream signaling pathways. A key distinction between the BCR and the TCRs is their interaction with antigenic epitopes. TCRs require that antigens be presented within the binding cleft of major histocompatibility complex or MHC molecules, while BCRs can engage with epitopes on free antigens or displayed on intact pathogens without MHC presentation. Additionally, TCRs are limited to recognizing protein epitopes, while BCRs can identify a wider variety of molecular classes, including proteins, polysaccharide, and lipopolysaccharides. Activation mechanism of B cells vary based on the type of antigen encountered. When presented with protein antigens, B cells act as antigen-presenting cells by displaying protein epitopes with MHC2 to helper T cells, classifying these antigen as T-dependent. In contrast, antigens such as polysaccharides and lipopolysaccharides can trigger B-cell activation directly without the need for T-cell involvement, categorizing them as T-independent antigens. This differentiation in activation pathway highlights the versatile nature of the B-cells in the immune response. The T-cell independent activation of the B-cell occurs when the B-cell receptors interact with T-independent antigens such as polysaccharide capsules and lipopolysaccharides. These antigens have repetitive epitopes and facilitate the cross-linking of multiple BCRs, providing the initial activation signal. Since helper T cells are not involved in this process, the second activation signal must originate from the alternative source, including interaction between the toll-like receptors and pathogen-associated molecular patterns or factors from complement system. Upon activation, B cells proliferate clonally and differentiate into plasma cells, which are specialized for antibody production. These plasma cells, devoid of surface BCRs, secrete large amount of pentameric IgM antibodies that are specific to the same antigen recognized by the original BCRs. The transformation marks a significant step in the immune response, enabling the body to produce antibodies rapidly in reaction to identified antigens. However, the T-cell independent response is generally short-lived and does not lead to the formation of memory B cells. As a result, there is no secondary immune responses upon subsequent exposure to T-independent antigens, limiting the effectiveness of this type of activation in providing long-term immunity. T-cell dependent activation of the B cell is a more intricate process than T-cell independent activation, resulting in a stronger immune response and the development of immunological memory. This activation can occur in response to either free protein antigens or protein antigen associated with an intact pathogen. When a naive mature B cell encounters a free protein antigen, it triggers internalization of the antigen. In contrast, when an antigen is a part of the pathogen, the B cell first extracts the antigen before internalizing it. Once inside, the protein antigen is processed and presented on MHC2 molecules, where it can be recognized by a helper T cell specific to the antigen. This interaction is known as linked recognition, is crucial for effective immune response. Upon activation through the linked recognition, Th2 cells secrete cytokines that stimulate the B cell proliferation into clonal daughter cells. These B cells undergo several rounds of division, and further cytokines form. Th2 cells encourage their differentiation into memory B cells and plasma cells. Memory B cells are primed to quickly respond to future exposures of the same protein epitope, while the plasma cells lose their membrane-bound B cell receptors and begin secreting the pentameric IgM antibodies. Following the initial production of the IgM, the Th2-derived cytokines from the plasma cells to switch from the IgM to other antibody classes, which is the IgG, IgA, or IgE, through a process known as class switching. This genetic rearrangement allows plasma cells derived from the same activated B cell to produce various antibody classes while retaining the same specificity of the original epitope. Thus, class switching enhances the versatility and effectiveness of the immune response, enabling a more tailored attack against the pathogens. T-cell dependent activation of the B cell is crucial for both primary and secondary adaptive immune responses. Upon the first encounter with a protein antigen, the T-cell dependent primary antibody response is initiated. This process begins with a lag period of approximately 10 days, sometimes we say 5 to 7 as well, during which antibodies are undetectable in the serum. This time is needed for various steps, including the binding of the antigens to the naive B cells, activation of the helper T cells, clonal proliferation of B cells. As the lag period concludes, the IgM levels of the serum rise due to the Th2 cells promoting the B cell differentiation into plasma cells. IgM peaks around 14 days post-exposure, while the IgG levels start to increase, reaching their peak in about 3 weeks in response. In contrast, the secondary immune response is more rapid and robust thanks to the memory B cells generated during the primary response. The lag period is shortened in just a few days, and the IgG production is significantly higher than the primary response. Moreover, antibodies produced in the secondary response exhibit greater effectiveness and affinity for their specific epitopes. Additionally, the plasma cells generated during the secondary response have a longer lifespan compared to those in the primary response, leading to sustained elevated levels of specific antibodies. This enhanced response underscores the efficiency of the adaptive immune system in recognizing and combating previously encountered antigens. This section will discuss the various kinds of artificial immunity, differentiate between the variolation and vaccination, and describe the different types of vaccine and explain their respective advantages and disadvantages. Vaccination is a highly effective strategy for disease prevention, representing a form of artificial immunity that stimulates the adaptive immune response. By mimicking a primary immune response, vaccines promote the production of memory cells, enabling the body to mount a robust secondary response when exposed to pathogens, all without experiencing an initial infection. This section will delve into the various types of artificial immunity, different vaccine forms, and the mechanism through which they induce protective responses. Adaptive immunity can be categorized into two main forms, active and passive immunity. Active immunity arises when an individual's own immune system is stimulated to defend against pathogens, while passive immunity involves the transfer of immunity from another organism. Both types can be further divided into natural and artificial categories depending on how the immunity is acquired. Natural active immunity develops through the direct exposure to a pathogen, leading to a lasting immune response. For example, recovering from a disease like a chickenpox or measles typically grants lifelong immunity. However, the duration of this immunity can vary significantly based on the specific pathogen and the nature of its antigen. Some pathogens may trigger long-term protection, while others may result in a shorter-lived immunity. Natural passive immunity is characterized by the transfer of antibodies from mother to the child, both prenatally and postnatally. The IgG antibodies crosses the placenta, providing essential protection to the fetus and lasting for up to 6 months after birth. Additionally, secretory IgAs are transferred through the breast milk, further supporting the infant's immune system during the early development. Artificial passive immunity involves the administration of antibodies from a donor to another individual, often for the preventive and therapeutic purposes. This method can be used to protect against diseases post-exposure, such as rabies or hepatitis, or to treat active infections. Historical examples include the treatment of Ebola patients with blood transfusions from recovered individuals, showcasing the effectiveness of this approach in reducing mortality rates. Artificial active immunity forms the basis of vaccination, where individuals are intentionally exposed to a weakened or inactivated pathogens or specific antigen to stimulate their immune response. This proactive approach aims to prepare the immune system for further encounters with the actual pathogens, ultimately enhancing protection and reducing the incidence of infectious diseases. To effectively control pathogen spread, a vaccine must induce protective immunity in a substantial portion of the population. While not everyone needs to be immunized, achieving a critical threshold is necessary to halt transmission. This concept, known as herd immunity, illustrates that the significant number of individuals are vaccinated. It creates a protective barrier around those who are unvaccinated. For instance, in a comparison between two elementary schools, one with a high vaccination rate and another with only half of the students vaccinated, the likelihood of an unvaccinated children contracting measles is considerably lower than the vaccinated school due to a presence of a greater number of immune individuals. Calculating the herd immunity threshold is specific to each pathogen and generally requires 80 to 90% of the population to achieve vaccine-induced immunity. This threshold is determined using the basic reproduction number, or R0, which indicates how many non-immune individuals could be infected by an infected person. For example, The herd immunity threshold for smallpox is approximately 80 to 85%, while for measles, which has a higher R0, it ranges from 93 to 95%. It is important to note that even with a high vaccination rate, such as 80%, the actual immunity level might be lower due to a varying vaccine effectiveness. This discrepancy underscores the challenges in achieving sufficient herd immunity, especially in larger uncontrolled populations where complacency or vaccine reluctance can further threaten public health. Variability in vaccine responses is common among outbred populations, with factors like age and health significantly influencing individual immunity. For instance, the influenza vaccine tends to be more effective in younger adults compared to the elderly. Weak immune responses can pose serious risks as they may not provide adequate protection against infections. Additionally, if individuals with poor immune system responses do contract the virus, there is a potential for the emergence of a vaccine escape mutant that can evade existing immune defenses. This phenomenon highlights the critical need to ensure high vaccination rates and effective public health strategies to mitigate the risk associated with the outbreaks. Herd immunity can help slow or stop a disease's spread, but without a vaccination, reaching the herd immunity by natural infection can be deadly if the mortality rate is high for the pathogen. In that cases, many people may die before the herd immunity can be reached by natural selection. Thousands of years ago, it was discovered that individuals who recovered from smallpox gained immunity to future infections. The practice of inoculation, known as a variolation, began in the 10th century China, where individuals were deliberately exposed to infectious material from smallpox scabs or pustules. This method involved injecting the material into the skin by administering it nasally. Most cases resulted in a milder infection, which ultimately provided individuals with protection against the more severe form of the disease. Despite its advantages, variolation was not without risk. Some patients experienced serious, even fatal infections. The contagious nature of the smallpox meant that variolation could potentially lead to outbreaks. Nevertheless, the practice gained popularity and expanded to regions such as India, Africa, and Europe, marking an important step in the history of disease prevention. Edward Jenner, an English physician, is often recognized for pioneering the modern vaccination process, building on the centuries-old practice of variolation. He observed that milkmaids who had contracted cowpox, a milder disease, seemed to be immune to smallpox. This observation led him to theorize that exposure to a less severe pathogen could offer protection against a more dangerous one. In 1796, Jenner tested the theory by injecting a young boy with material taken from a cowpox lesion, making a significant moment in the medical history. The boy exhibited mild symptoms, including low-grade fever and discomfort, but notably did not develop smallpox when later exposed to the virus. This led to the term vaccination, derived from the Latin word for cow vacca, highlighting the use of the cowpox in the prevention of smallpox. Modern understanding reveals that the cowpox virus shares a genetic and antigenic similarities with the variola virus responsible for smallpox, meaning the exposure to the cowpox helped the immune system recognize and respond effectively to smallpox. The success of Jenner's smallpox vaccine spurred further advancement in immunization, inspiring scientists like Louis Pasteur to develop vaccines for various diseases, including rabies, cholera, and anthrax. Over the 20th and the 21st centuries, the field of vaccination expanded significantly, leading to the creation of effective vaccines for the multitude of viral and bacterial diseases such as chickenpox, hepatitis, measles, and tetanus. This ongoing work in immunology has profoundly impacted public health, reducing the incidence of many once common diseases. Live attenuated vaccines utilize a weakened strain of pathogen to induce the subclinical infection, aiming to activate the adaptive immune system. The process of attenuation involves reducing the pathogen's virulence through methods like genetic manipulation, which remove the key virulence factor, or through a prolonged culturing in unnatural hosts or environments. These strategies promote mutations that diminish the pathogen's ability to cause disease. By establishing an active yet mild infection, live attenuated vaccine evoke a more robust immune response compared to other vaccine types. They engage both cellular and humoral immunity while promoting the formation of immune memory, which leads to long-lasting protection. Interestingly, vaccination of one individual can sometimes result in the natural transmission of the attenuated pathogens to others, triggering similar immune responses in those individuals. However, live attenuated vaccines present certain disadvantages. The challenges of long-term storage and transport can complicate their use. Additionally, patients, especially those who are immunocompromised, may experience symptoms of a disease due to the active infection. Another concern is the potential for the attenuated pathogens to revert to the original virulent form. Inactivated vaccine consists of whole pathogens that have been killed or rendered inactive through heat, chemicals, or radiation. For these vaccines to be effective, it is crucial that the inactivation process preserves the integrity of the key antigens on the pathogen, allowing the immune system to recognize and respond appropriately. Since inactivated vaccines do not induce an active infection, the immune response they elicit is generally weaker and less comprehensive than that elicited by live attenuated vaccines. Typically, the response is limited to humoral immunity, and there is no risk of transmission to others. Furthermore, inactivated vaccines often require higher doses and multiple booster shots, which can lead to inflammatory reactions at injection site. Despite these limitations, inactivated vaccines offer advantages such as stability for long-term storage and ease of transport. They also pose no risk of causing severe active infections. However, side effects can still occur, reminding patients of need for careful monitoring. Subunit vaccines. Subunit vaccines differ from live attenuated and inactivated vaccines by exposing individuals only to key antigens of a pathogen rather than whole cells or viruses. These vaccines can be created chemically degrading a pathogen to isolate its essential antigens, or through genetic engineering to produce these antigens directly. The focus on essential antigen means that the subunit vaccines generally have a lower risk of side effects compared to other vaccine types. They stimulate a targeted immune response while avoiding unnecessary exposure to the entire pathogen. This makes them a safer option for many individuals, particularly those with compromised immune systems. Subunit vaccines are particularly valuable in providing protection without the risk associated with the live or inactivated pathogens. The efficiency and the safety of these vaccines make them an important tool in public health. Toxoid vaccines are designed to prevent diseases by causing a bacterial toxins by using inactivated toxin known as toxoids. Rather than introducing whole pathogens to the patient, these vaccines primarily activate humoral immunity, which neutralizes the toxin responsible for the disease. They are particularly important in preventing illnesses where bacterial toxins play a significant role in pathogenesis. Conjugate vaccines are representative of a specialized form of subunit vaccines that combine a protein with a capsule polysaccharide. The combination enhances the efficacy of the vaccine against pathogens that use protective polysaccharide capsules to evade the immune system, leading to severe infections like meningitis. While traditional subunit vaccines may not elicit a strong immune response in children under 2, conjugate vaccines effectively stimulate antibody production by linking T-dependent protein antigens to the polysaccharide capsule. This dual approach encourages a robust immune response against both components. There are 5 basic approaches of producing vaccines. Each uses components of pathogenic virus that a vaccine is intended to target. A vaccine developer may produce large quantities of the virus of interest and chemically inactivate it, which is the inactivated vaccine. Attenuate it, pathogenicity through laboratory manipulation, replication-competent attenuated vaccine. Produce individual proteins free of a viral nucleic acid in a subunit vaccine. Molecularly clone all or portion of the viral genome for preparation of recombinant DNA vaccine, which is known as a recombinant vaccine, or design mRNAs to produce viral antigens and delivered through nanoparticles like an mRNA vaccine. The methods are designed to adhere to the principle that Pasteur would understand. The most common commercially successful vaccine simply comprise attenuated or inactivated virus particles. The next two slides will discuss the novel and recent science to make cheaper, cleverer, or more effective strategies that are about to take over the age-old art of vaccine making. A DNA vaccine is a type of vaccine that transfects a specific antigen-coding DNA sequence into the cells of an organism as a mechanism to induce an immune response. DNA vaccine work by injecting genetically engineered plasmid containing the DNA sequence encoding the antigens against which an immune response is sought to the cells directly produce the antigen, thus causing a protective immunological response. DNA vaccines have theoretical advantages over conventional vaccines, including the ability to induce wider range of types of immune response. Introduction of DNA vaccines into a patient leads to the uptake of the recombinant plasmid by some of the patient's cells, following by transcription and translation of antigen and presentation of these antigens with MHC1 to activate adaptive immunity. This results in stimulation of both humoral and cellular immunity without the risk of active disease associated with live attenuated vaccines. Some DNA vaccines are already in use. Several DNA vaccines have been tested for veterinary use. In some cases, protection from disease in animals has been obtained. In others, not. 2005, a DNA vaccine against West Nile virus was approved for the use in horses in the United States. Canada also approved a DNA vaccine to protect fish from infectious hematopoietic necrosis virus. A DNA vaccine against the Japanese encephalitis virus was approved for the use in humans in 2010 in Australia. Research is ongoing over approaches for the viral, bacterial, and parasitic diseases in humans, as well as for cancers. In August 2021, Indian authorities have given emergency approval of a ZyCoV-D developed by Cadila Healthcare. It is the first DNA vaccine approved for humans for COVID-19. An mRNA vaccine is a novel type of vaccine that utilizes messenger RNAs to trigger an immune response by delivering antigen encoding mRNA into cells. The vaccine instructs them to produce a foreign protein that typically associated with pathogens like viruses or cancer cells. These processes stimulates an adaptive immune response, enabling the body to recognize and eliminate these harmful entities. The mRNA is encapsulated in a lipid nanoparticle which protect the RNA strands and facilitate their entry into the cells. In terms of reactogenicity, the mRNA vaccines exhibit similar adverse reaction profiles to traditional vaccines, with some individuals prone to autoimmune responses potentially experiencing adverse effect. However, mRNA vaccines offer several advantages over conventional ones, including faster design and production, lower cost, and capability of inducing both cellular and humoral immunity without interacting with the genomic DNA, while certain mRNA vaccines, such as the Pfizer-BioNTech COVID-19 vaccine, require ultra-cold storage. Others, like the Moderna vaccine, do not have stringent storage requirements. The making of the mRNA vaccine. First, the mRNA in vitro transcription. Using DNA with an antigen encoding sequence from the pathogen as a template, mRNA in vitro transcription products contain a single-stranded RNA or a double-stranded RNA. The ssRNA structure normally includes a 5' cap, 5' untranslated region, open reading frames, and then 3' untranslated region and a poly tail structure. To enhance the stability and detection by the mRNA by RNA detecting TLRs in the cells, instead of uridine, a modified nucleoside, pseudo-uridine is used during the in vitro transcription of mRNA. In 2023, the Nobel Prize in Physiology and Medicine was awarded to Katalin Karikó and Drew Weissman of University of Pennsylvania for their discoveries concerning modified nucleosides that enable the development of effective mRNA vaccine against COVID-19. As a side note, in 1997, Katalin Karikó met Drew Weissman, an immunology professor at the University of Pennsylvania, and they began collaborating. with Weissman's funding playing a crucial role in advancing her research. The scientific community was skeptical about the use of nucleoside pseudoiridine in a vaccine and whether that would enhance the stability in the cell. Their combined expertise in immunology and biochemistry proved highly effective as they tackled various challenges, ultimately gaining recognition for their work. Weissman remarked on the difficulties they faced, stating, "We had to fight the entire way," while also highlighting Kariko's exceptional persistence in navigating the challenges of academic research. B. RNA translation and antigen presentation. Through endocytosis, mRNA enter the cytoplasm. Some mRNAs combine with the ribosome of the host cell and translate successfully. Antigen proteins can be degraded to antigenic peptides by proteasome in the cytoplasm and presented to cytotoxic T lymphocyte via major histocompatibility complex 1 pathway, or they can be released out of the host cell and taken up by the dendritic cells. Then they degraded and presented to helper T cells and B cells via MHC2 pathway. B cells can also recognize released antigen proteins. C. Self-adjuvant effect. Various pattern recognition receptors can recognize the mRNA in vitro transcription product. Single-stranded RNA can be recognized by endosomal innate immune response like TLR7, TLR8. Double-stranded RNA can be recognized by endosomal innate immune receptors like TLR3, and cytoplasmic innate immune receptors like protein kinase R, retinoic acid inducible gene 1 protein or RIG1, and melanoma differentiating associated protein 5 like MDF5, 2,5' oligoadenylate synthetase or OAS. Based on these, the mRNA products can stimulate the secretion of pro-inflammatory cytokines and type 1 interferon, which leads to antigen-presenting cells activation and inflammatory reaction. However, they can also activate antiviral enzymes that cause stalled mRNA translation and mRNA degradation. Self-explanatory slide showing the prominent vaccine against the SARS-CoV-2 or COVID-19 vaccines. The highlighted vaccines are either approved or approved for emergency usage. BioNTech is a biotechnology company based in Mainz, Germany. They partnered with the pharmaceutical giant Pfizer. The reason for this is the cost of running the clinical trials. Smaller pharmaceutical companies can't invest $500 million or more US dollars to run the clinical trials, so they have to partner with a bigger company which can sponsor it. Moderna is also a smaller company but receives $483 million federal assistance from BARDA to run its clinical trial. Just a fact that by now the US government has paid Moderna $6 billion US dollar for the vaccines. The J&J vaccine is developed by Janssen Pharmaceuticals in a Belgian pharmaceutical company, which is a part of the much bigger company Johnson & Johnson, but even Johnson & Johnson received $454 million from BARDA to run its clinical trial. It is currently discontinued though. Several other available vaccines are also mentioned on the slide. How the COVID-19 vaccines could be developed so fast? The mRNA delivery technology via liposomes, lipid nanoparticle, existed from the late 1970s to transfect animal cells in laboratories for gene expression. In late 1990s, Dr. Katalin Kariko and Dr. Drew Weissman of the University of Pennsylvania developed the pseudoiridine-based mRNA vaccine strategy that would increase their half-life in a cell when delivered, as they would recognize by the TLRs slower than the foreign RNA with regular iridine. Dr. Kariko co-founded an mRNA vaccine startup, RNARX, where she served as a CEO from 2006 to 2013. From 2013 to 2022, she was associated with BioNTech's RNA Pharmaceuticals, first as a vice president and promoted to senior vice president in 2019. BioNTech RNA Pharmaceuticals were already involved in developing mRNA-based vaccines, so the stage for implementation was already set years ago before the arrival of COVID-19. Adenovirus technology has been there for years. There are a dozen of adenovirus-based veterinary vaccines, just no human vaccines. In recent history, there has been no demand to develop a vaccine. That's why no one has actively run any clinical trials, and it was not necessary. So the above-mentioned monetary facts are the keys to vaccine development. To develop a vaccine, you need these astronomical amounts of money, and the subjects who can get infected. Both were available for COVID-19 and explained how these vaccines were developed so fast. In contrast, it took 10 years to develop the Ebola vaccine. Why? Unfortunately, it was not the threat to the wealthy countries, and no company wanted to invest $500 million for a vaccine that would never be paid for. The company would not make a profit. In addition, as the Ebola outbreaks were in the pockets of a few African countries, getting 40,000 clinical trial volunteer subjects was also very difficult, which was not the case for COVID-19. Now, this course will allow you to explain these common questions asked by the public all the time. This is the same analogy. During the world wars, the countries became industrialized at lightning speed, but the countries that did not take part in the world wars became the third world developing countries. So it is always the necessity is the mother of invention. These are your links to learning. Take a look at some of these videos. Some of them are really fascinating, particularly the Qatar incident about the polio vaccines that destroyed the trust in healthcare, and you will know why it is. And this is a book that is suggested, really nicely written by Paul Offit. It's about Maurice Hilbert. He created 9 vaccines, and every child receives all of these vaccines like mumps, rubella, and largely forgotten. And Paul Offit became friend with Hillman in his final months of his life and conducted extensive interviews. So this is basically the story of Maurice Hilbert. Thank you.